So what we're going to talk about today will be an extension of what we talked about Friday, and of course anything we cover today is fair game for the exam on Wednesday. And so what we talked about last time was hormone actions, and we talked about hormone effects, and we talked about the importance of the hormone receptor complex formation. Well, we're going to extend that story today into particular kinds of hormones. We're going to talk about the details of G-protein coupled receptors and how they bind hormones on the outside of the cell and communicate across the phospholipid bilayer to affect cell functions on the cytoplasmic side. So we'll spend a lot of time on that, and then we'll talk a little bit about the effects of the hydrophobic hormones, particularly the so-called steroid hormones. But the other major idea that we need to get through today is the hypothalamic pituitary axis. This is one of the major hormonal axes in the body, and it basically is a connection from the brain, the hypothalamus, to the pituitary. Now we're going to see the pituitary is broken into two regions. The posterior pituitary is an extension of the hypothalamus itself, and neurons in the hypothalamus will go right into, extend into the posterior pituitary and release neurotransmitters from cells in the posterior pituitary into the blood. The anterior pituitary is a different creature. The anterior pituitary arises from epithelial cells embryologically in the pharynx of the developing animal. Those cells then will migrate during development from the pharynx all the way up and sit right beneath the brain. And this now becomes a new structure. That new structure is called the anterior pituitary. And that anterior pituitary now is going to play a really, really important role in communicating to the rest of the body. But its only connection to the hypothalamus is through the blood vascular system, and we'll talk about that. But as we work our way through this, we're going to see first that there's a hormone receptor complex formation, and the first one we're going to talk about are the large hydrophilic hormones. The large hydrophilic hormones we talked about last time are going to bind to the extracellular side of transmembrane proteins. There are a number of these. Now, the ones that you may run into in another class are the receptor tyrosine kinase receptors. We're not going to say anything about those. What we're going to talk about are the so-called G-protein coupled receptors, sometimes known as GPCRs. The G-protein coupled receptors traverse the membrane 7 times. These are the so-called 7-pass transmembrane proteins. Something like 4% of the genome is dedicated to making these G-protein coupled receptors. They're really, really important receptors in the central nervous system, the peripheral nervous system. They're all over, and they're important in regulating cell function. So we're going to emphasize the regulation of cell function here through the binding of a hormone on the extracellular side, communicating across the membrane to a cascade of events. Fundamentally, what we're going to see is the 7-pass transmembrane protein binds a hormone on the outside of the cell, but that generates a structural or conformational change in this 7-pass protein, which now permits the activation of something known as a Gtp binding protein. We're going to see the Gtp binding protein is composed of 3 separate proteins, an alpha, a beta, and a gamma subunit. For our purposes in here, the alpha subunit is going to be the main player. We're going to see that when the alpha subunit has a guanosine triphosphate bound to it, it's active. And an active alpha subunit with GTP now can turn on, what we're going to talk about here, can turn on something called adenylyl cyclase. Adenylyl cyclase then will activate a protein kinase that will phosphorylate the proteins, phosphorylate other proteins, and we'll walk our way through that. Then we're going to talk a little bit about hydrophobic hormones. Now, the hydrophobic hormones that we're going to emphasize here will be the steroid hormones. We touched on those last time. But recognize, too, that the thyroid hormones are also hydrophobic hormones. As such, those hydrophobic hormones don't require an extracellular binding site. They can skitter right through the phospholipid bilayer, get into the inside of the cell, and on the inside of the cell, in the cytoplasm, they can now bind to a cytoplasmic receptor. And we'll see that these cytoplasmic receptors now, once bound to the hormone, are going to allow the receptor and the hormone to enter the nucleus. And once inside the nucleus, the hormone receptor complex now affects genes as a transcription factor. So through this process, we're actually going to turn on genes. And then we'll talk about the hypothalamic pituitary axis, the posterior pituitary being an extension of the hypothalamus, releasing these 2 hormones, oxytocin and vasopressin, that'll hold you to, and then the anterior pituitary, which is a different set of cells. This is separate from the brain. The only communication between the anterior pituitary and the hypothalamus is through the hypothalamic portal blood system. So blood will flow through the hypothalamus, deliver that blood to the anterior pituitary. But that blood, after passing through the hypothalamus, will be carrying a set of hormones called releasing hormones that will bind to troph cells, that release stimulating hormones that then go to the target neuron, or target gland rather, that will then feed the information back. And we'll talk about the so-called negative feedback. But let's begin here with a reminder of what hormones do. And you'll recall that they affect enzyme activity. You'll recall that they affect channel opening and closing. You recall that they affect exocytosis, and that they can affect gene transcription. Let's start here with the story about the large hydrophilic hormones. These are typically protein or peptide hormones. The particular example that we're going to use here is something known as a G protein coupled receptor. It's often abbreviated as the GPCR, G protein coupled receptor. We mentioned this last time. We emphasized the idea that if we have the impenetrable barrier of the phospholipid bilayer, the communication from outside to inside had to occur through a transmembrane protein. That these large hydrophilic molecules simply couldn't get through the couldn't get through the hydrophobic tails of the phospholipid bilayers. The one we're going to talk about here is the G protein coupled receptor. Whenever you see this term, think 7-pass transmembrane protein. I'm going to simplify this a bit. We did this last time too. There's a little U out here, comes across the membrane. 1, 2, 3, 4, 5, 6, and 7. Where this is the amino terminus, and this is the carboxy terminus. Now this is clearly oversimplified. We simply have this splayed out transmembrane protein. But recognize that these extracellular regions and the cytoplasmic regions that are down in the cytoplasm, that those are out in the aqueous environment so they can form a three-dimensional shape. I'm going to simplify this and say that the hormone binds out here in the cup. But in reality, recognize that all of these extracellular loops are involved in making a three-dimensional shape that will accept and provide the specificity and affinity for the hormone. But since hormone specificity and affinity were from Friday, let's now talk about how once we attain the specificity of the hormone binding to the receptor, I can't emphasize too much that hormone function depends on the hormone receptor. Hormones can't do anything without binding to the appropriate receptor. So in this case, this hormone binds to this receptor. in ways we don't quite understand, the binding of the hormone to the receptor changes the three-dimensional structure of the cytoplasmic region of the protein. So we change the structure here, but mostly we're going to see that the change in cytoplasmic structure of the G protein coupled receptor permits binding of an activated Gtp binding protein. Well, the next character in this play is going to be this Gtp binding protein. The Gtp binding protein is composed of three subunits. The Gtp binding protein has an alpha subunit, it contains a beta subunit, and it contains a gamma subunit. Because there are these three subunits, this is said to be a hetero, means they're different, trimeric, means there are three, Gtp binding protein. So here are the three subunits, alpha, beta, gamma. Normally, the beta and the gamma and the alpha all live together. They're all happily bound one to the other. But for these guys to all live together happily, this alpha subunit has to have attached down here a Gtp. This is guanosine. Guanosine triphosphate. Diphosphate. Now, when the Gtp is bound to the alpha, it's inactive. Now, the binding of the hormone to the receptor allows this alpha subunit with the Gtp attached. I'll try to put a squiggle in here to make this a little easier to see. The conformational change allows this alpha subunit to bind to the cytoplasmic side of this G protein coupled receptor. So in other words, the change in shape of the cytoplasmic side of this G protein coupled receptor allows for this alpha subunit to come and bind to the cytoplasmic side of this protein. The binding of the alpha subunit with this Gtp attached to this protein causes another enzyme to come along and get rid of the Gtp and replace it with guanosine triphosphate. Now remember, I said when this was alpha, beta, gamma, everybody was all together. This had Gtp, this wasn't active. Now, everything changes at this point. When this alpha subunit exchanges its Gtp for a Gtp, it tells the beta and gamma to go suck eggs. Go away. And the alpha subunit with the Gtp attached now goes off on its own. It goes off on its own in the phospholipid bilayer. So now, at this point, we have the boot, its alpha subunit, and it has a Gtp attached to it. So what I want to do now in the upper panel is describe the cascade of events. Following hormone binding to a G protein coupled receptor. Now, if you get down into the weeds of this, and we'll actually address this a little later when we talk about the cardiovascular system, you'll recognize that there isn't simply one G protein coupled receptor. That there are some G protein coupled receptors that are excitatory, there's some G protein coupled receptors that are inhibitory, and there's some that release calcium from internal stores. They do a whole bevy of things. The one we're going to talk about here is one known as G alpha S. This means the alpha subunit is stimulatory. I can even say it's stimulatory to activating an enzyme known as adenylyl cyclase. So in this sort of imaginary cell that we're going to draw as a box, we'll use the same nomenclature we have above, where this structure is the G protein coupled receptor. When it binds the hormone here, that causes a cytoplasmic change in its configuration. The change in the configuration permits the alpha subunit to sidle in along with its beta and gamma. Under these circumstances, this starts out with a GDP. But then the binding of the alpha subunit to the cytoplasmic region of the G protein coupled receptor causes the GDP to leave and be replaced by a GTP. Then the alpha subunit comes free with its GTP attached. The major target for this alpha subunit with the GTP attached is a transmembrane protein over here that goes across the membrane 1, 2, 3, 4, 5, 6, has an active region down here. 1, 2, 3, 4, 5, 6 has an active region down here. This is called adenylyl, A-D-E-N-Y-L-Y-L cyclase. What adenylyl cyclase does, it's an enzyme that converts adenosine triphosphate, ATP, into something known as cyclic AMP. I'll draw this out quickly and I'll show you a slide in a moment, but again, here is our 5-carbon structure, 1, 2, 3, 4, and 5. This is a ribose, so down here it has a hydroxyl, but it also has hydroxyl, the number 3 carbon. The number 5 carbon out here initially is ATP, so it's carbon, oxygen, phosphorus, oxygen, phosphorus, oxygen, phosphorus. We've seen this pattern before when we form the phosphodiester linkages between adjacent deoxyribose and ribose sugars. We're going to modify this story just a little bit, and rather than linking one sugar to the next, we're going to link this number 5 carbon through the oxygen to a phosphorus to an oxygen bond. So this is now cyclic AMP. Bond. That's cyclic AMP. It's been cyclized. Simply been cyclized by linking the number 3 oxygen to the number 5 oxygen to form that phosphodiester linkage. But that now becomes a really important character because cyclic AMP the generation of cyclic AMP from this adenylyl cyclase. The adenylyl cyclase has taken ATP and made cyclic AMP. This cyclic AMP now becomes an actor, and it now turns on a protein called protein kinase A. Now, an important idea I want you to hold on to, because we'll refer to this over and over again, is whenever you see this term kinase in biology. Kinase is an enzyme that adds phosphate groups to proteins. To help you hold on to this, the only place we can add these phosphates, for this enzyme at least, is to threonines and serine. And what makes these amino acids special is they have hydroxyl group on them. We can use that hydroxyl group to make a phosphate linkage. So now a protein kinase A has this ability. We can say the protein kinase A phosphorylates proteins. And whenever you add this enormous phosphate group to a protein, you alter the protein functioning. So you may remember, I'm going to do it here. So you may remember that the hormone receptor complex can alter enzyme activity. Well, here we are. We have altered the activity of the adenylyl cyclase. So this is one of the hallmarks of all hormone functions. We have used the binding of the hormone to the receptor to alter adenylyl cyclase activity. So that means we've altered enzyme activity. We can alter ion channel activity. Well, there are transmembrane proteins out here. There are transmembrane proteins out here that are in fact channels. These could be sodium channels, they could be potassium channels. Commonly, they're calcium channels, but this is an ion channel. And when we add these great bulky phosphate groups to the channel, we alter channel activity. So we can either change the probability of opening, we can make it more permeable to an ion, we can make it less permeable to an ion. We alter channel activity. Similarly, those phosphorylation events can affect exo and endocytosis. And then finally, the other thing the hormone receptor complex can do is it can feed into the nucleus, where it will affect gene transcription and translation. The next thing we're going to do is we're going to talk a little bit more detail about this gene transcription and translation story, particularly from the point of view of the hydrophobic hormones. But before we go on to that, there's a major concept or idea I want to emphasize from what we have here. And this is the idea of amplification. You can see that the binding here of a single hormone molecule to its appropriate receptor can now turn on maybe 10 or 20 or more of these alpha subunits of the alpha beta gamma trimeric Gtp binding protein. So now where we've bound one hormone molecule to the receptor, we've now activated multiple alpha subunits, which can now in turn activate maybe another 100 of these adenylyl cyclase molecules. They can now produce maybe 1,000 or 10,000 of the cyclic AMP molecules that can then in turn activate maybe 10,000 or 100,000 protein kinase A molecules that can feed back onto thousands upon thousands of channels. So the idea here is the importance of these G-protein coupled receptors functioning as a cascade of events that ultimately leads to this tremendous amount of amplification. So any questions to there then? If you put your phone away, I'd appreciate it. Thank you. Yes?
So protein kinase A only phosphorylates threonine and serine?
So protein kinase A is one of those kinases called a serine threonine kinase. If you get into another cell biology course, you're going to find out there are a whole series of kinases called tyrosine kinases. We're not even going to touch on those. The tyrosine kinase is particularly well known for affecting gene transcription and translation.
What were the circles or adenyl cyclic?
Oh, I just added more detail than I needed. These little circles are meant to be the active regions of the adenyl cyclase. So they have this complex transmembrane regions, but there are two regions within the cytoplasm that are the active portions of the enzyme. So it takes just a minute here. This first image is simply showing a pretty picture of the G-protein coupled receptor as it traverses the membrane 7 times. And I won't belabor it too much, but here's the amino terminus, here's the carboxy terminus, and there are many thousands of these G-protein coupled receptors. And out here is the region on the on the extracellular side, and these extracellular regions will form a 3-dimensional shape to provide specificity and affinity for a large hydrophilic hormone. Then when the binding of the large hydrophilic hormone occurs, that changes the shape here on the cytoplasmic side. And on the cytoplasmic side, the change of shape permits the binding of the G-protein, Gtp binding protein rather. So again, here's the G-protein coupled receptor. It binds the hormone. That now permits the interaction of the alpha, beta, and gamma subunits. Normally, the alpha, beta, and gamma subunits are sitting there. Each of these is connected to the inner leaflet of the phospholipid bilayer by a fatty acid side chain. So they're happily floating around in here. Then normally the alpha subunit has a GTP attached. But when this alpha, beta, gamma subunit sidles up to an activated G-protein coupled receptor, then the alpha, beta, and gamma are all living together, but now something changes. The guanylyl exchange factor comes along, clips loose the GDP, replaces with GTP, and the alpha subunit basically tells the beta and gamma, I don't need you anymore, go away. This alpha subunit then goes out and turns on adenyl cyclase. Now, inaccurately, this is shown as only going halfway across the membrane, but in reality, it should cross the membrane many times. But the most important idea here is that with this alpha subunit and its GTP attached, that will activate adenyl cyclase to convert ATP into cyclic AMP that activates protein kinase A that phosphorylates proteins. Then the next image is simply what I drew on the board with a much prettier picture, of course. Here's the adenosine triphosphate. This is the formation of the cyclic bond, that is adenyl cyclase, takes the triphosphate, clips off 2 of the phosphates. The remaining phosphate now is linked from the number 5 carbon to the number 3 carbon. This is the cyclizing effect. Then we can break this back into cyclic AMP by using a phosphodiesterase. So any questions on that?
Is the alpha S, alpha I referring to the alpha subunit?
So the question here is over here on the introduction, where we talked about G alpha S, G alpha I, and G alpha Q. Some of these alpha subunits are stimulatory. This stimulates adenyl cyclase, so this is a GS. There are other varieties of these alpha subunits will actually turn off adenyl cyclase. They'll downregulate adenyl cyclase. That's GI for inhibitory. Sometimes you'll see these referred to as GOs. And then finally, there's GQ. Why it got Q name, I have no idea. But the Q, in fact, releases calcium from internal stores. Cell biology class will spend a lot of time on that. But here we're just going to say there are G-protein coupled receptors. They turn on an alpha subunit by exchanging a GDP for a GTP activated that turns on adenyl cyclase to make cyclic A. That's the second messenger. that then leads to these downstream effects, particularly the phosphorylation event. Anything else? Yes? So kinase A phosphorylates ion channels, among other things. And one of the mysteries here, I don't think still has been solved, is how you take G protein coupled receptors that are something like 4% of the genome, which seem to do these generic things. They end up phosphorylating everybody and their brother. And how do you manage to get that to be controlled at a cellular level? And we think that probably has to do with neighborhoods, that we put all these cascade events in fairly narrow regions of a cell. And so that only is affecting those narrow regions. But that's a bit of that's conjecture and hypothesis more than anything. Anything else? If not, let's now take this story, leave the upper panel. We're going to take this story and think about the other kind of signaling. The other kind of signaling that is carried out by the hydrophobic hormones. So the emphasis here was on the hydrophilic hormones and their requirement to use a transmembrane protein for downstream signaling. But what we want to think about now is the classic effects of the hydrophobic hormones. And we mentioned last time that the two of the really important hydrophobic hormones were the thyroid hormones and the steroid hormones. We're not going to really differentiate these here. If you get into another physiology class or cell biology class, you'll find particularly that those thyroid hormones go directly to the nucleus, whereas the steroid hormones come through the cytoplasm first. And that's the story we're going to emphasize here. We're going to tell the story, for the most part, from the point of view of hydrophobic steroid hormones. Hydrophobic steroid hormone effects on genes. So again, here's our cell. This is the phospholipid bilayer. This is plasma membrane. This is the nucleus. Now, I'll remind you here, and I won't draw it out in too much detail, but I'll remind you that the steroid hormones are based on cholesterol. And that the cholesterol is this 5-membered ring. A, B, C, and D. I'm sorry, 4-membered ring, A, B, C, and D. But because of that flat, plate-like nature, and because it's predominantly hydrophobic, then it will be carried in the blood on carrier proteins. But its function is inside the cell. That these hydrophobic hormones don't require an extracellular receptor site. These guys, because they're flat and lipophilic, can go across the phospholipid bilayer and into the cytoplasm. And we find then in the cytoplasm these steroid hormone receptors. And except for a few details, because the thyroid hormones are also hydrophobic, the same story applies. But this sort of rectangle now, I'm going to label as a steroid hormone receptor. The steroid hormone receptor is a cytoplasmic protein. Of course, as a cytoplasmic protein, you should immediately think that it's going to be synthesized on free ribosomes. if we lay this thing out, as with all proteins, there's an amino end and a carboxy end. Toward the carboxy end of the molecule, down here, is a region of the protein known as the hormone binding domain. So if we were to extend that to here, inside the cell, in the cytoplasm, the hormone will bind at this point. So this is the hormone binding domain. It will bind out here. I won't draw it too much, but so that's where the hormone binds. But if you remember, one of the hallmarks of these steroid hormones is that these guys function as transcription factors. If you're going to function as a transcription factor, you've got to bind to DNA. So where does that tell us this molecule is headed? It's got to go to the nucleus. And so the next region I'm going to put here in orange is said to be a nuclear axis. A nuclear access domain. I think of these as the keys to open the portcullis of the nuclear pore. So this will bind to the nuclear pore, tell the nuclear pore to open, and now allow for this molecule to go into the nucleus through those nuclear pores. So now we have addressed this molecule with the hormone attached to the right spot, but it doesn't do us any good unless there is a region of this protein which binds to DNA, and that's this region. This is the so-called DNA binding domain. So the hormone has bound to the receptor in the cytoplasm. It's used its nuclear access domain to open the door to get into the nucleus. And then once in the nucleus, it has to turn on a particular set of genes. So to turn on a particular set of genes, it needs to find the appropriate promoter regions to which it can attach. Once this DNA binding region binds to the portion of the genes that it wants to turn on, that then turns on this region out here. I'll put in green, called the transactivation domain. The transactivation domain effectively recruits the proteins of the transcription factors. You'll remember that whenever we talked about a transcription factor, we said it was a whole group of, maybe as many as 15 proteins. This is now one of the proteins that's added to that cohort. And then once the entire cohort is assembled with the receptor, its hormone, and the rest of the transcription factors, this then, of course, recruits RNA polymerase to transcribe genes. So in other words, the binding of the hormone in the cytoplasm leads to moving this whole complex to the nucleus. And because of the sequence of amino acids in the DNA binding domain, it will bind to a particular promoter. Once bound to the promoter, then this region, the transactivation domain, helps recruit the rest of the proteins, the transcription factor, to recruit RNA polymerase, and in this way we can turn on certain genes. So those cells that contain these receptors in the cytoplasm will turn on those genes. But if those cells have not made any of those receptors, they will be unaffected by the hormones that may enter their cytoplasm. So any questions on that? So be able to compare and contrast these two mechanisms. One being across the phospholipid bilayer through G protein coupled receptors, the other the cytoplasmic receptors for hydrophobic hormones. Yes?
Just two questions. One, so is there a protein that's binding to the nuclear axis domain to take it into the...
So is there a protein that's binding to the nuclear axis domain that brings it in? Not as far as I know. My understanding is that the binding of the hydrophobic hormone out here to the hormone binding domain changes the shape just enough that this opens it up. Now, in fact, I don't know if I want to say this, it takes two of these. And one of the things that the hormone binding to the hormone binding receptor does is it recruits another one, and they have to dimerize. And once they dimerize, that opens up the nuclear binding domain, the nuclear axis domain that gets you in.
This is all one transcription factor?
This is a portion of the transcription factor. Remember, transcription factors typically 13 to 15 proteins, and this is only one member of that whole cohort. Yes? Does each hormone have its own specific steroid hormone receptor? Yes, that's true. And in fact, it's one of the truisms that I would like you to take away from this, in that the way a cell responds to a hormone is dependent entirely upon its receptor. It's the receptor that determines the function of the hormone. It's not the hormone itself. It's the receptor it binds to. We better move on to the hypothalamic pituitary axis. So let's go to the upper panel, and we'll lay out the hypothalamic pituitary axis and think about the anatomy and the hormonal flow in this system. And the characters in this play are going to pretty much be about four different characters. The first character is going to be part of the brain, called the hypothalamus. The second character is going to be an extension of the hypothalamus, called the posterior pituitary. The third character is going to be an independent set of epithelially derived cells, called the anterior pituitary. Then the fourth character in the play is going to be the target hormone. So first we'll lay these out as anatomical structures. And then we'll think about the interplay among them. And finally finish with a discussion of something known as negative feedback. But we'll start here by labeling this panel as the hypothalamic pituitary axis. Now we'll begin here, and I'm going to put the structure in. Label this the hypothalamus. This structure is meant to be the posterior pituitary. And even as the way I've drawn it, the posterior pituitary is an extension of the hypothalamus. There are neurons in the hypothalamus. I'll put these in green. There are neurons up here in the hypothalamus. They send processes or axons out into the hypothalamus. They send processes from the hypothalamus into the posterior pituitary. And then there will be blood vessels here. in the posterior pituitary. So this is a capillary. And these neurons now release either oxytocin or vasopressin. Vasopressin is important for kidney function. And oxytocin is important for smooth muscle contraction. We'll run into these again when we talk about the kidneys. We'll run into oxytocin when we talk about when Dr. Bauma is here talking about reproduction, because oxytocin is important in uterine muscle contractions. But that's all part of the brain. There's no question, but that's part of the brain. But now there's another structure here, and I'll put this in orange. This structure I'm going to label as the anterior pituitary. Embryologically, these cells were part of the epithelium of the pharynx. And during embryological development, those cells have migrated in the pharynx and tucked themselves up in this region right beneath the hypothalamus, right next door to the posterior pituitary. But these are not part of the brain. These are separate cells entirely. Within the anterior pituitary, there's a cell type. These are called troph cells. These troph cells are in the anterior pituitary. In the hypothalamus, up here, are neuroendocrine cells. These neuroendocrine cells release or secrete molecules known as releasing hormones. The only connection between the anterior pituitary here and the hypothalamus is through a blood vascular system. There are a series of blood vessels here known as the hypothalamic, pituitary, portal vasculature. So that in blue, the releasing hormones are released into the blood. So these are releasing hormones. They are released into the blood, but the releasing hormones, the receptors for releasing hormones, are on troph cells. So the receptors for the releasing hormones are down here on the troph cells. And in response to the releasing hormones, the troph cells release stimulating hormones. And the target for the stimulating hormones is the so-called target gland. In the last minute, let me lay something out for you. And this is an important idea. Let me deep six this. And this is a kind of four-box model of this system. So to begin with, these are the releasing hormones from neuroendocrine cells in the hypothalamus. Then in orange, in the orange box here, what we have are troph cells. These are the troph cells of the anterior pituitary. And these are releasing or secreting, stimulating hormones whose target is the target gland. Then the target gland releases its hormone, and this hormone then feeds back on the anterior pituitary, feeds back onto the hypothalamus, and these are inhibitory. And what we're talking about here now is the concept of negative feedback. And you'll need to know that for the exam. And I'll leave you with that.